People ask me, why should I care about biochemistry? And being the kind of person who sometimes likes to answer questions with questions, I say, how does coffee wake us up? Why do yeast make alcohol? Why do I check my phone 50 times a day? Why do I get sleepy? How come plants don't get sunburn? Why don't elephants get cancer? Why do all animals age and yet bacteria do not? I like biochemistry because it gives answers that are eye-opening. Not only do you learn things you didn't expect, you also get to learn a powerful way of asking and answering questions. That's how biochemistry helps design life-saving treatments, improve the food supply, and answer questions in areas spanning from archaeology to criminal justice. In fact, there are few areas of science today where biochemistry hasn't had some impact. Biochemistry is new as the sciences go. Separately, the subjects of chemistry and biology have been around since the time of the ancient Greeks and Egyptians. But the idea of combining them is much more recent. 200 years ago, there was no evidence that a science of biochemistry was even possible. Even 100 years ago, the word biochemistry was barely coming into use, and the field had scarcely begun. And yet, biochemistry, this young science, has already unraveled chemical pathways and processes in cells that make us who we are. Personalized medicine is becoming possible thanks to disciplines biochemistry has spawned, including genomics and metabolomics. Biochemistry is truly what I like to call the science of us. This science of us is a single grand story that is much bigger than all of humanity. And it's much more exciting than anything else in chemistry. Organic chemistry, for example, has many aspects to explore, but it lacks the very thing that brings people to biochemistry, the bio part. And yet, this more exciting version of chemistry is entirely grounded in the same rules as the rest of chemistry. Biochemistry brings together the best of biology with the best of chemistry. Does it get any better? The earliest roots of biochemistry date from almost 2 centuries ago, when an experiment by Friedrich Wöhler accidentally created a compound we now know as urea. It turned out to be identical to a crystal that appeared when he dried urine. This demonstration made it clear for the very first time that ordinary chemistry had to be possible inside of cells. As early investigators began to take apart the cell, they increasingly began to discover both the magic of biochemistry and its roots in traditional chemistry. 2 discoveries at roughly the same time in the 1860s were inheritance and the existence of DNA. But the significance of these remained unrealized for decades. The famous physicist Erwin Schrödinger wrote a seminal book for lay readers entitled What is Life?, based on lectures he had been giving in Dublin in the 1940s. This book set the theoretical framework for what we now take for granted, that everything we associate with life has its roots in molecules. Watson and Crick in 1953 pointed to Schrödinger's book as inspiration for their search to find the structure of DNA. Everyone knew, of course, that the chemistry of reproduction had to be distinctive, but it took a long time to realize that what enabled cells to reproduce was their ability to store information, read that information, and then reproduce that information. No chemistry anybody had ever seen before had done that. Classical biochemistry had been concerned with enzymes and other molecular reactions found inside and among cells. Now a new field called molecular biology came into being with the master instructions that cells need to carry out their activities. The 2 fields became 2 sides of the same subject. Which name to use for this combined subject depends on where we start, so we'll call it biochemistry. And while biochemistry might sound like a subject only experts could handle, the truth is that there are some nice simplifications right here at the start. Take the periodic table, which has well over 100 elements. With only a few exceptions, biochemistry is not concerned with those individual atoms at all, much less their internal structure of positively charged protons and neutrally charged neutrons. Biochemistry is mostly about molecules. Moreover, the molecules of biochemistry are overwhelmingly built using primarily just 6 bonding elements. They are carbon, oxygen, nitrogen, and hydrogen, with supporting help from sulfur and phosphorus. What matters for joining these atoms together is electrons. Chemical bonds always involve electrons. What's pretty remarkable about that is the electrons are the smallest parts of their atoms by a long ways. Electrons are negatively charged and located outside the nucleus of the atom. The number and kinds of bonds an atom can make is due to the number of electrons it can share, release, or steal. Of those, the bonds we care the most about in biochemistry involve sharing of electrons. It is these covalent bonds that stick us together. Cells are mostly water, which is just hydrogen covalently bonded with oxygen, dihydrogen oxide. The oxygen can make 2 bonds. Each hydrogen can only make 1 bond. But the more important fact is they share electrons with each other, with oxygen kind of hogging them and hydrogen getting the slightly shorter end of the stick. The other 4 elements that are of primary importance for making bonds in chemistry also like to share. The element whose atoms are arguably the most important for life is carbon. Carbon's importance is directly traced to its electrons. It has 4 electrons that are involved in reactions, and they all participate in sharing with other atoms. Sometimes the sharing is 50-50 equal, and sometimes it may be a bit unequal, but carbon never gives up its electrons entirely or takes those of another atom. Carbon's ability to make 4 bonds also makes it central to the construction of big and complicated molecules. A carbohydrate molecule, for example, is a bunch of carbons that have been hydrated with a bunch of water. Biochemistry, as you will see, is all about big carbon-centered molecules and their relationships to the water of the cell. Let me show you a convention for depicting how molecules are stuck together. In beginning chemistry, we might write letters or even full names for every single atom with a short line in between to show the bond. Water is 2 hydrogen atoms linked to oxygen. 2 atoms of oxygen come together to make molecular oxygen by making 2 bonds. Double bonds are shown by 2 lines between atoms. Carbon molecules can have all their carbons written out, but we can make the same diagram simpler if the carbon atoms are not explicitly shown. Instead, the angled lines are used, and the carbon is assumed to be at the meeting point of angled lines depicting the bonds. In such diagrams, hydrogens attached to the carbons are also omitted entirely. These simplifications help a lot when showing bigger carbon molecules. For example, here's a fatty acid drawn both ways. Besides carbon, hydrogen, and oxygen, the 4th most abundant element in our bodies and the most abundant in our atmosphere is nitrogen. Nitrogen can make 3 bonds with its electrons. When nitrogen bonds to hydrogen, as it commonly does, it forms what we call an amine, which is related to ammonia. The feel-good molecule dopamine has an amine group at one end. Amines also give their name to amino acids, the simplest of which is glycine. Amino acids are the building blocks of proteins, and proteins are the stars of biochemistry. A 5th element that is important in many proteins is sulfur, as you know from the smell of eggs when their proteins start to break down. Sulfur can make 2 bonds and is found in 2 of the amino acids. Cysteine is the amino acid you'll hear the most about thanks to its sulfur, which is fairly reactive. The 6th element important for building biomolecules is phosphorus. In cells, we pretty much see phosphorus only when bound to 4 oxygen atoms to create a unit known as phosphate. Now, phosphates are important for storing energy and for being on-off switches that control whether a protein is on the job or off-duty. The energy to power cells comes from a triphosphate called ATP. Phosphates are also part of the backbone of the structure of DNA as well. I sometimes like to refer to the atoms of these 6 elements together with the contrived word sponge. If you want to build biomolecules, it's easy. All you need is sponge. Sponge is really all you need. There are some other players in biochemistry. Many of these are atoms and molecules we refer to as minerals. 8 minerals are most abundant in biochemistry: sodium, potassium, magnesium, calcium, iron, zinc, copper, and chlorine. Minerals fall in a different category from sponge because they do not usually share electrons when they form bonds. Chlorine steals electrons. Each of the other minerals is a supplier of electrons. In the watery environment of the body, such molecules are broken up and become charged ions. Our bodies are not made of ions. Instead, we make use of ions. Stealing electrons involves gaining a negative electric charge, while supplying electrons means a loss of negative charges by the atom participating. Since atoms start out with a charge of 0, we can tell the number of electrons an atom has gained or lost by its charge. Sodium, for example, is a supplier of 1 electron, so it typically has a charge of +1. And chlorine steals electrons, so it has a charge of -1. Put them together and you make sodium chloride, known as table salt. But in the watery environment of our bodies, the ionic bonds holding the salt are broken, and we use the ions. Iron and copper are 2 ions with some additional abilities. We find them in cells with 2 possible charges each. For iron, it can exist as iron +2 or as iron +3. Copper can exist as copper +1 or copper +2. What makes this so interesting is the ability of iron and copper to flip between the 2 states differing by 1 electron turns out to be very important for handling energy in cells. Copper plays 2 important roles in the body. One involves energy generation. It helps in respiration by transferring electrons to oxygen to make water. And this turns out to be the reason you breathe. No copper would certainly take your breath away. The second role of copper is in helping to protect cells from random damage actually caused by the oxygen that you breathe. The process of making an ion is called ionization. Ions are very important to cells. When minerals dissolve in water, they form ions and literally come apart. Sodium chloride in your salt shaker splits into sodium ions and chloride ions when you dissolve them in water. Since water is 70% of the weight of cells, interactions with water in the cell are crucially important. Ions are not building blocks of cells, but ions perform critical tasks in and around the cells of our body. Individual atom ions like sodium and potassium are very tiny compared to proteins and other giant cell molecules with thousands of atoms. The tiny size of these ions allows cells to regulate their movement using nanoscopic protein channels specific for each one. And their tiny size allows them to move very rapidly, while their electrical charge gives them important roles in the electrical circuits of our nerve cells. It is partly thanks to the small and charged characteristics of these mineral ions that nerve cells can communicate so rapidly in the body. Meanwhile, proteins are what I call the workhorses of the cell. Now, when I say protein, that might make you think of foods like steak or fish or eggs. But the reason we need dietary protein is very important. We digest those proteins to obtain their amino acids, and we use those amino acids to build our own proteins. Proteins are polymers, which are long strings of molecules joined end to end to end to end to end. There's more than one way to make a polymer, though. Non-living polymers tend to repeat a single building block over and over. Plastic polymers are like this. Plastics have the same building block molecule repeated over and over. What such repetition provides is uniformity. Look at a piece of plastic and it will appear the same no matter what part of it you examine. If the Borg of Star Trek were a single polymer, this is the kind of polymer they might be. By contrast, protein polymers are much more lively, we might say, with much more individuality and personality. Proteins do not use a single building block repeated monotonously over and over. Instead, proteins have at their disposal and often use 20 different building blocks, all amino acids. We say the proteins are assembled from building blocks because assembly implies a plan. A plan is needed because each of the thousands of proteins found in cells has a unique order of building blocks joined to each other. And unlike all other molecules, the plan for each of our proteins comes direct from a cell's DNA, as written in the genetic code. And that sequence of building blocks determines the shape of the protein and what a protein does. Many proteins are quite large, often with over 1,000 amino acids in a single protein molecule. That's why we call them macromolecules. Now we're getting into the magic. I said proteins are the workhorses of cells, and I want to give an example that shows us a lot about chemistry inside of cells. Some proteins speed up reactions occurring inside of cells. A huge family of proteins responsible for speeding up all sorts of reactions are called enzymes. Speeding reactions is cool, but there are non-protein molecules that speed reactions too. We call them catalysts. The platinum in the catalytic converter of your car is an example. What makes enzymes almost magical is their incredible speed. Some enzymes in the cell speed reactions more than 1 trillion times faster than a catalyst like platinum can speed the catalytic converter reactions in your car. In fact, one of the secrets of life is that cells rely on enormously speeded up reactions. In the absence of enzymes, some reactions can take millions of years to occur all by themselves. Millions of years? We don't really have that long, right? So the speed of enzymes is crucial. Moreover, enzymes are not only faster than the fastest race car, enzymes are also very picky. They are more particular about their partners than Peter Piper picking a peck of pickled peppers. We call this high specificity. So enzymes are a bit like a glove attempting to fit on a hand. If it does not fit, no reaction on it. Platinum as a catalyst is not that way. Platinum can catalyze the reactions of many molecules, but what it gains in numbers of different reactions it can catalyze, it loses in the speed of those reactions, sometimes by factors of trillions. How do enzymes act so fast and so specifically? This comes back to the protein polymer and the building blocks. The specificity of an enzyme is rooted in the very specific arrangement of the building blocks in its polymer. Change the order of amino acids and you change the shape of the enzyme. Shape of the enzyme turns out to be one of the most important considerations in biochemistry. A basic principle is that structure determines function. Enzymes have highly specific shapes, and those specific shapes only allow molecules with corresponding shapes to fit in them. Fitting into enzymes in a very specific way is critical because that's where the enzymes catalyze reactions. If a molecule can't fit into an enzyme, its reaction will merely grind along at the speed of non-life. And this doesn't mean that merely a handful of reactions in cells are sped up. The vast majority of biomolecules found in cells get made in reactions catalyzed by enzymes in that cell. Remember, each enzyme catalyzes reactions quite specifically on only one or a handful of different molecules. So any given cell must have thousands of enzymes to catalyze the thousands of different reactions in that one cell. And that's exactly what cells have. Let's talk more about what it means to be a cell. Cells are what we call the fundamental unit of life. Remember, each cell has thousands of enzymes speeding up thousands of highly specific reactions. In scale, we need a microscope to observe cells, but cells in turn would need their own microscope to get down to see the level of molecular activity going on inside. Overall, we could think of cells as the houses for the molecular reactions we've been describing. Like houses, cells have surrounding walls in the form of a membrane. Some cellular houses are more like tents, with a simple design, smaller than houses, and containing only a single room for the reactions to live in. Bacterial cells have single rooms. We call them prokaryotes. With no internal walls, bacterial houses keep their possessions in the same room. Most other cells, like the ones in our body, are more like houses or apartments subdivided into rooms, each room with a function. Rooms in cells are called organelles. There are several, including a nucleus, where the DNA is stored, and mitochondria, where energy is made. In plants, there are also chloroplasts, where energy from the light of the sun is captured in photosynthesis. And the outer membrane of plants is wrapped in another layer, actually called the cell wall. Cells with the organizational scheme of a house or apartment are called eukaryotes. Many eukaryotes, whether plants or animals, are multicellular and sort of like cities containing many houses. However, a few eukaryotes, such as yeast and amoebas, happily maintain the rural life of a little house on the prairie. But whether it's a tent, a house, or a vast city of many houses, one of the remarkable things about the biochemistry of cells is this: all living things are remarkably similar. The molecules they contain are similar. The reactions their molecules undergo are similar. And the ways they store information inside themselves are similar. In fact, the degree of similarity is almost shocking. If we look at how a simple one-celled bacterium breaks down sugar to get energy and compare it to how that happens in a human being, we see that the reactions are identical. Petunias break down sugar the same way as poodles and people. Whether it's the breakdown and synthesis of fat, the storing of chemical energy, or the synthesis of proteins, all cells take the same basic approach. Now, there are, to be sure, a few differences between cells. In fact, drug makers work hard to exploit minor differences between us and bacteria when designing antibacterial drugs against infections. But the similarities between cells far outnumber the differences. For us, learning biochemistry, this similarity provides a great simplification. When you learn the biochemistry for one organism, you've already learned a great deal about every other organism. We don't have to learn one biochemistry for our pets or pests and a different one for ourselves. These deep similarities are what make it possible for us to use bacteria, yeast, and animals as biological factories to make human proteins, such as insulin, for medical use. Now that we've surveyed the big picture, let's look at some of the simplest molecular building blocks. These are the functional groups covered in any organic chemistry course. But here again, what we need for biochemistry is much simplified. What we're doing is a little like learning to read the symbols on a map. Carbon is the basis for all the nomenclature I'm going to give you here. If our molecule has carbon bonded only with hydrogen, that makes a hydrocarbon. The simplest hydrocarbon is methane, a waste product where a single carbon makes bonds with 4 hydrogens. Methane is produced by Daisy and Bessie as they graze happily in the fields. Methane is such a common byproduct of cellular respiration that scientists seeking evidence of life on other planets search for it in the atmosphere. Added to another molecule, methane gives up one of its 4 hydrogens and is called methyl. Methylation of your DNA can affect whether or not proteins are made from it. A ring of carbons is benzene. A ring of carbons with 3 double bonds is phenyl. If we have carbon bonded to oxygen and hydrogen, many more biological compounds are possible, including alcohols, aldehydes, ketones, and acids. Alcohols are the simplest, with just an OH group. The thing to remember is that this OH group is called a hydroxyl group. The word is put together just like the atoms, with the YL included, just like methyl, to show it's attached to something else, hydroxyl. Ethanol with 2 carbons and the hydroxyl group is the alcohol found in beer, wine, and spirits. Although in everyday speech we use alcohol to refer only to ethanol, in chemistry the word refers to all molecules with a hydroxyl group linked to a carbon. Often there's an OL in the name. Cholesterol, for example, has a hydroxyl group. Dopamine, which besides its amine group also has 2 hydroxyls. And coming in with 3 hydroxyls is glycerol. This is the molecule that combines with fatty acids to make any of the fats. If an OH hydroxyl group is bonded to a phenyl ring, then the phenyl ring is called a phenol. Serotonin, the happiness molecule, is a phenol. There are also polyphenols by the hundreds found in plants that give us antioxidants and other exciting nutrients whose importance is increasingly understood. When carbon makes a double bond with an oxygen, that's a carbonyl group, also known as an aldehyde. The vitamin A in your eyes that gives you vision is an aldehyde called retinal. Acetaldehyde is what your body makes if you drink alcohol and causes the misery known as a hangover. Notice that it has a carbon double bond with oxygen, with the double bond at the end of the molecule. If the location of the double bond is in the middle of the molecule instead of the end, then we call it a ketone. The so-called keto diet, which is very high in fat, causes your body to make ketones. One of these, called acetone, has 3 carbons, with the middle one double bonded to oxygen. Acetone is also found in nail polish remover. Many sugars are ketones. We also see ketones as intermediates in the breakdown and synthesis of fatty acids in cells. Carbon can also have the double bond of an aldehyde and the hydroxyl of an alcohol attached to the same carbon at the end of the molecule. That's all it takes to make a carboxylic acid. Acetic acid, which is notable for giving vinegar its tang, is the poster child. Notice the C double bond oxygen and the OH, which we call a COOH group, or a carboxyl. Anything with a carboxyl group goes under the general category of carboxylic acids. When we talk about an acid in biochemistry, we're also usually talking about these carboxyl groups with a COOH group. These are weak acids, but the fact that they don't go all the way is what lets them act as buffers to help stabilize acid levels in cells. And we have a lot of acids built this way. We have bile acids that help with the digestion process. All the fatty acids have a carboxyl group. For example, myristic acid found in coconut oil is 13 carbons plus a carboxyl. All the amino acids get the acid part of their name from having a carboxyl group. Let's also take it up a notch and see what happens when a carboxylic acid reacts with an alcohol. The resulting molecules are called esters, with a molecule of water released in their formation. Simple esters often have a fruity smell. The smell of bananas, for example, comes from combining acetic acid with an alcohol called isoamyl alcohol to make a compound called isoamyl acetate. The glycerol we just met has 3 hydroxyls just waiting. So when 3 fatty acids react with glycerol, the triple ester that results is a fat. This one molecular process, called esterification, is what makes all the fats that help food taste so much better, everything from olive oil to butter. Now, don't worry about terminology. The language of biochemistry is very powerful, and I use terms to help you know what we're talking about. But when I mention any of these terms again, I'll remind you about them. When new terms appear, I'll introduce them. You don't have to memorize names to understand what's going on here. That's because this isn't a course about names, equations, memorizing, or any of the standard things that people stereotypically associate with science teaching. I've designed this to be a one-of-a-kind course where you can enjoy biochemistry while you learn it. And I truly want you to enjoy it. I work hard to do things differently, and this course is something I'm proud of. I hope you enjoy it. Learning biochemistry is easier than other parts of chemistry in several ways. Biochemists don't have to deal with the ins and outs of over 100 elements. Cells have picked the very best ones for us, and becoming familiar with only a small subset of reactions from organic chemistry lets us focus on things that really matter, the biology that this chemistry gives rise to. What also helps is that anything that's more complicated in biochemistry is also more exciting. Often this is the biochemistry you hear reported in the news. New medical treatments, new insights about health and wellness, new discoveries about the foods we eat. Biochemistry lets you understand life, your own and all life around you. What's wonderful is how much you can understand for yourself. Coming up next, why water is the most important biological molecule of all.